Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type your questions in for our speaker in the question and answer window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions. Your questions in the question and answer window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Eric Isberg from Savillex.
Thank you very much. So for today's presentation, I'm going to give a quick introduction to Savillex. I'm going to talk a little bit about fluoropolymer containers, and then show 4 different case studies highlighting the benefits of fluoropolymer containers for vaccines and regenerative medicine applications. So for the bioprocess market, Savillex is focused on PFA, FEP, and ETFE fluoropolymer bottles and vials. We have 2 adjacent manufacturing facilities here in Eden Prairie, Minnesota. One is dedicated exclusively to our ready-to-use, or what we call RTU products. We, as a company, were the first to stretch blow mold fluoropolymer bottles, and we are the only supplier of ready-to-use fluoropolymer bottles and vials to the bioprocess market. Fluoropolymers have several advantages: extremely high purity, very low extractables, thermal stability and a very wide temperature range, and unmatched durability and essentially unbreakable during usage. 2 popular applications for our bottles are vaccines and regenerative medicine. Both require high purity, chemically inert containers. Both involve storage and shipment of final product. And in the case of regenerative medicine, include contact with live cell cultures. Now, some of the issues with hydrocarbon polymer containers is that they have the danger of a very high extractable profile. Traditional polymers like polycarb, polystyrene, polypropylene, and polyethylene can have relatively high chemical extractable levels. And high extractable levels have been shown to inhibit cell viability and reduce final product stability. So what are the risks of using common polymers? Lower process yield due to cell inhibition and death, a risk of chemical contamination of your final product, reduced product stability, and these problems are magnified as the product becomes more concentrated. So what I'd like to do now is ask a poll question. Now I can't see the poll question, I apologize. But how important is the consideration of leachables to your process? If you could please answer the questions. I'm actually going to show you the results of the survey. Very interesting. Most people consider it to be important, if not critical, that extractables be managed and detected in their product containers. So there is a better way with fluoropolymers. Fluoropolymer bottles have extremely low extractables and provide a better environment for cells, higher product yields, higher stability of your final product, and better patient outcomes. So now I'm going to move into the case study section of the presentation. Our first case study is a vaccine that's manufactured in the US in bulk, and then that vaccine is shipped over to a second European facility for the formulation and filling process. So our PFA bottles were chosen due to durability during the shipping as compared to, say, bags, which do tend to be prone to punctures and tearing and other failure modes. PFA was chosen by this customer due to a very, very low extraction profile. And in this case, this product is provided as an assembly. And so you can take a look at the picture I'm showing on this slide. The bottle on the right-hand side is actually the assembly that the customer uses for this process. So they batch the vaccine bulk aseptically using a sterile connector into a vented 1 liter or 2 liter PFA bottle. The second case study is a more recent one. This is lipid manufactured in Europe and then shipped to a second European site. And it's actually used in the mRNA COVID vaccine manufacture. As most people know, lipids are a very critical component to the mRNA COVID vaccine manufacturing. Bottles, again, were chosen due to the durability during shipping. And the bottles were also chosen due to inertness with not only the lipid itself, as well as a low extractions profile, but also the lipids were stored in a 60 to 80% ethanol base. So they were concerned about extractables. They were concerned about, in their case, leachables coming out into the product during shipping. In this case, this was supplied as a ready-to-use product. And it was well accepted by the customer because of the ready-to-use format. They were in a very aggressive schedule. We need the product yesterday type schedule. And so we were able to meet that because we had this process all set up and that we were easily able to convert them over to the ready-to-use product. The third vaccine case study is farther up in the process. So this is a customer that needed a custom engineered vial. They had up to 15 milliliters of culture going into their vaccine manufacturing process. And the existing cryovials that they had been using in other parts of the process were simply too small. Most cryovials are in the 0 to maybe 5 mil range. Maybe you can get some as large as 10 mils, but we were able to design one out of ETFE fluoropolymer. ETFE is nice in that it's gamma stable. And in this case, they wanted a gamma stable product because the other components coming into their process were also sterilized using gamma. So it allowed them to have a consistent sterilization process throughout all their raw materials. This was also, again, supplied, ready to use, clean, and pre-sterilized. I also here have some results of a tech note that we just published just this week, showing the temperature mapping of our full line of PFA and ETFE bottles. And you can see the results by bottle size. I've just personally published this on my LinkedIn account. So those of you that are linked into me, feel free to go on there and take a look at it. Otherwise, you can go right onto the Savillex website, and the URL is shown here on the slide. The third case study is regenerative medicine. So in this case, this was an FDA-approved cartilage graft, and it's stored at a 2 to 8 degree temperature range, so it's relatively benign storage. But they were using polycarbonate bottles, and what they found was that they had significant loss of that graft over the shipping process, which is only a 1 to 2 day shipping process. So the viability of their graft was dropping pretty significantly. And they were convinced that it was a combination of the shape of the container as well as the fact that it was made from polycarbonate. So they reached out to us. We were able to supply them with our 100 mil PFA bottle, again, as a pre-sterilized product. And they successfully did their shipping viability and container closure testing of our product, and just recently received approval from the FDA to switch over to our containers. So we're pretty excited about this product. This is our direct-to-patient. This is a product that's actually used in surgical processes. So we're very proud of this. So I hope you enjoyed the presentation today. If you want to learn anything more about Savillex or our Purelex products, feel free to reach out to me or visit our website as shown here. Thank you very much.
Great. Thanks, Eric. So the first question is, how are fluoropolymer bottles sterilized?
That's a good question. So PFA and FEP fluoropolymer bottles are typically sterilized using autoclave sterilization. The ETFE product is unique in that it is sterilized using gamma. So those are the two main methods used for sterilization of fluoropolymer products.
Okay. And what is the range of sizes that are offered?
So the range of sizes that we have in our fluoropolymer product line, we have vials down to very small sizes, 500 microns all the way up to even 60 milliliters. Our bottles range in size from 50 milliliters up to 2 liters.
Okay. And what testing is performed to analyze extractables, and how do you determine the level of extractables?
So that's another good question. We in this industry typically follow what they call the BPOG protocol. We're in a unique situation in that fluoropolymers are actually used to contain or to hold the control samples used in the BPOG protocol. So Savillex vials, for example, are used to hold the control samples. So we most frequently will do customer specific combination leachables extractables study. So for the regenerative medicine customer, we did a protocol that was very specific to their process. It gave very good data, again, showing the very, very low extractable levels of our product.
Okay. And how do you secure after sterilization the cap closure to keep the product sterile?
So most of our customers, they receive their product with the container and the closure packaged together in a double bag, so they're separate. Once they fill the bottle, then they will attach the closure. And we have adapters that then can attach to the closure, and you can use a torque wrench. And we have a series of torque specifications for each of our products based on application. So, for example, the torque specification for a bottle that's going to go into liquid nitrogen would have a higher torque spec than one that would be used at 2 to 8 degrees. But we validate those torque specs, and then every lot of bottles that we manufacture, we actually do our own version of container closure integrity testing, where we attach the closure at the suitable torque, and then we do a pressure decay, actually a hydrostatic pressure decay test.
Okay. So it looks like we have time for just one question. If we didn't get to your question, they will be sent to Eric and he can follow up with you directly. So please continue to type your questions in to the question and answer window. How do you ensure the bottles are clean prior to sterilization?
So we actually manufacture the bottles in the Class 7 clean room. So the entire stretch blow mold equipment is actually located in the clean room. And we package the bottles immediately off the press. So the bottles are still warm. The operator takes the bottle, puts a closure on it immediately, inspects the bottle, and then packages it immediately. So it's a very clean process. The vials are done in a very similar fashion, where the vials are injection molded. Right off of the injection molding press, there's automation that then picks those parts, and it goes down a conveyor that's again covered in a Class 7 clean room environment, and then the vials come out and are immediately packaged again within that Class 7 clean room. We also test all of the vials and bottles for USP 788 particles, bioburden, and endotoxin. So every lot gets tested for those three. So we assure that that cleanliness is maintained.
Okay, well, thank you, Eric. And thanks to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive an email providing you with a follow-up link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.
Thank you.